Antibody modelling. Aims and objectives then for this session are to refresh a little bit about antibody structure, to refresh the idea of comparative modelling, and to compare and contrast general comparative modelling with antibody modelling. So before we start, why are antibodies so interesting? Well, around a third of drugs in development are antibodies. At least a quarter of drugs in phase 2 or later clinical trials are antibodies. Half of the top 10 grossing drugs are antibodies. And the reason for all this is that antibodies can function in different ways. They can be used simply for binding, just like a normal drug does, so they can act as inhibitors, they can act as agonists and antagonists by binding to receptors, and they can act simply to block interactions. They can also trigger the immune system to kill cells. They can also be used for targeting drugs or radioactivity. They can be used for cross-linking, and importantly, they can bind to targets that are simply not possible with small molecule drugs, because small molecule drugs need to bind into small pockets to make enough interactions to have enough affinity and specificity. Because antibodies have a large binding surface, they don't need to do that. Why then are we interested in antibody modelling? Well, first of all, because of humanization. This is the process where we take the loops that form the binding site from typically a mouse antibody or maybe a rat or a rabbit, and then graft that onto a human framework. But to do that and to get it to bind, we often need to make additional changes in the framework from human back to mouse or rat or rabbit or whatever it was, in order to get good binding. And in order to select those residues, some of the methods require you to have a structure of the antibody to analyse and decide which residues you're going to change. We also use antibody modelling to look at structural features, so things like patches of unusual residues on the surface, which might be immunogenic, or patches of hydrophobic residues that might cause aggregation. And thirdly, understanding antibody-antigen interactions allows us to enhance or modify the binding. Part 1, Introduction and Alignment. So hopefully you will recall from the first year lecture that comparative modelling is the process of building a three-dimensional model of a protein based on the known structure of a homologous protein. But antibodies are somewhat different. For a start, we have many structures available. Well over 1,000 different antibodies have their structures solved. And of course, they're all homologs. Secondly, we only need to worry about modelling the FV fragment of an antibody. That's the variable domains. The rest is constant, and therefore we don't have to worry about modelling it. Third, the FV fragment is highly structurally conserved, despite the fact that it is variable. It's only generally the CDRs that differ. And even these are relatively conserved in structure, apart from CDRH3. So really the problem comes down to modelling just 6 loops, and of those 5 are relatively easy, and it's only the 6th one that's difficult. So just to remind you of the domain structure of antibodies, we have 2 heavy chains shown in blue and yellow, and the 2 light chains both shown in red, coming together to form this Y-shaped structure. And the bit that we're interested in modelling then is the VH in blue and yellow, and the VL in red here and here. So the rest of the structure is formed of constant domains, and they are constant in both sequence and in structure. There are different classes and subclasses of antibodies where these things differ a little bit, but of course structures of all those have been solved. And of course occasionally you do get allelic variations, but those are generally just 1 or 2 amino acid differences. So this cartoon version of the same image shows you the domain labels. We have the 2 domains in the light chain, 4 domains in the heavy chain. IgM and IgE are different as they have an additional domain which actually replaces the hinge in here. So it's the size of one of these other domains, but it comes in here instead of this flexible hinge. So all we're interested in is really modelling this FV fragment, the VH and VL domains, and that really boils down to just modelling these CDR loops that form the binding site. Zooming in on the structure, here we have the FV fragment containing the VH and VL domains interacting here with an antigen. This happens to be an antibody from a mouse called HIHEL5, which is binding to lysozyme. Now, as we saw in the previous first year lecture on protein modelling in general, proteins can have very high structural similarity despite the fact that their sequence similarity is very low. So in this case we're looking at hemoglobin and myoglobin. They have only 27.7% sequence identity, yet their structures are virtually identical. You can see a few little differences here, for example in this loop up here and this region around the terminus here, that's a little bit different, but really the rest of the structure is very conserved. Now in the case of antibodies, it's loops like this that actually matter, because it's the loops that form the binding site. Before we continue, we just make a couple of definitions. The first of these is the target, that's the sequence or structure to be modelled, and normally we have a target sequence but no target structure. Then we have what's variously known as either the parent or the template, and this is the sequence and structure that's used as a basis for building a model. Now, in classical comparative modelling, there are 8 steps. The first of which is to identify the templates or parents that we're going to use. And then the second is to align the target sequence with the parents. Once we've done that, we can go on to the actual process of modelling. And the first step of that is to identify structurally conserved regions. So these are the regions that in all instances look pretty much the same, and the structurally variable regions, which are generally loops, and as we saw in the hemoglobin myoglobin example, you can see some differences in those regions. We then go on at step 4 to inherit the structurally conserved regions from the parent or parents. We build the structurally variable regions. We model in the side chains where those have changed from the parent structure to the target. We refine the model and evaluate errors. And at that stage we may cycle round again from the evaluation step to modifying the alignment and repeating this process. There's also the method known as modeler, which has the first steps being the same and the evaluation of errors being the same, but replaces all these in-between steps with essentially a version of molecular dynamics where we take constraints from the parents. These days, of course, there are other methods as well. Things like AlphaFold 2 has been in the news before Christmas, and that's a method that uses machine learning to predict the distances between amino acids in order to predict the overall structure. From the perspective of modelling antibodies, people are generally using something close to this classical 7-step plus evaluation approach. So I'm going to describe the first 2 of those steps now and make the comparison with the standard approach to modelling of normal proteins rather than antibodies as we go along. So the first step is identifying the parents, and normally that's just done by searching the target sequence against sequences from the protein databank using something like FASTA or BLAST. With antibodies, we have specialised databases of antibody structures. So, for example, we have one called SACS, which is Summary of Antibody Crystal Structures, which is automatically updated every month. There's SABDAB, which comes from a group in Oxford, and there's ABDB, which is also our resource, which is derived from SACS and provides you with pre-numbered structures. It deals with issues such as having multiple copies of an antibody in the same PDB file, or multiple different antibodies all bound to one antigen in the same PDB file, and sorts out all these sorts of problems. So despite the fact that we have these databases of antibody structures, we still need to find the best parent or parents to use. And there are different ways of doing that. We could base it just on sequence identity, but then we have to choose, are we going to look at the sequence identity just across the framework or across the CDRs as well? We could also take into account what's known as canonical classes of the CDRs, and we'll come back to that in a few minutes. But this is the conformations of the CDRs. So if we can actually choose a parent structure which has the right conformations, or approximately the right conformations of the CDRs, as well as of the framework, then that's obviously going to give us some sort of advantage. Now, in general, in modelling, alignment is the most critical stage. But it's also often the most difficult stage to get right, because the correct alignment is the structural alignment rather than purely the sequence alignment. So in other words, if you had the two structures, fitted them to one another in 3 dimensions, and then read off the sequence alignment, that's the alignment that we're aiming for. Now, if you only have sequences, or at least you have one sequence which has a structure and one sequence which doesn't, and you perform a sequence alignment, that may not be the same as the structural alignment. Now, in the case of antibodies, the FV, as we've seen, consists of a framework which supports the CDR loops. And I've shown that here with the CDR loops shown in colour, and there are also these additional loops at the binding site end of the domain, but these are not hypervariable in sequence and aren't normally part of the binding site. Now, with one or two small exceptions, the only insertions and deletions that we see in antibodies are in the CDR loops. And consequently, the alignment generally across the frameworks is absolutely trivial, and we just have to concentrate on the alignment in the loops. Even here, though, the alignment is much simpler. As I said, there are generally no insertions and deletions or indels in the frameworks, and for the loops, all we need to do is use one of the structure-based standard numbering schemes that are available for antibodies in order to imply the alignment. So here we're looking at about 8 different structures of antibody CDR-L1 loops, and you can see they vary in length from this one in dark blue, which is 10 residues long, up to this one in red, which is 17 residues long. But the fact that we have a standardized numbering scheme, where this is 24, 25, 26, 27, 28, 29, 30, means that if we apply that numbering scheme, we know that the insertions are going to occur in this region, and that structurally variable part of the loop is reflected in the numbering. So this is residue 30, this is residue 31, and here is 32. In this case, there is no residue 30, it's a shorter loop. So we skip residue 30, and this position is somewhere between 30 and 31. In the other ones, there are insertions between residue 30 and 31, and that's indicated by giving them letters like 30A, 30B, 30C and so on. So here I've taken a set of 8 different antibody CDR-L1 loops, and I have aligned them using a multiple sequence alignment. And you can see that really these are all over the place in terms of the alignment. So we would be, if we use this as the basis for building a model, we'd be putting gaps down here and one insertion here and one insertion there and a gap here and so on. If we apply the standard numbering scheme to these sequences, this is what we end up with, and we end up with all the gaps in the standard positions here, which correlates with where the insertion is in the loop. So we've covered choosing a framework, and we've covered aligning the sequence of the target with the parent, and shown that really all you need to do is apply the standard numbering scheme and nothing else. We don't need to use normal alignment programs. In the second part, we will go on to look at how we start to build an actual model.